So on this slide here, we have kingdom protista, and we are going to discuss about the kingdom protista. Let's start. Under the kingdom protista, we have general characteristics that all the organisms under this kingdom protista are having. So we are going to start from that point before we discuss some examples of organisms that are found under this kingdom protista. So let's start. Number one, they are eukaryotic, and karyotic means nucleus. That means they have a true nucleus, a nucleus that is being surrounded by a membrane. Number two, they are microscopic. That means you cannot see them with your naked eyes. And they are single cell organisms. That means they are unicellular. Unicellular organisms, these are organisms that are made up of only one cell. Number three is that some are free living. That means they cannot cause disease. Example, Eglena. While some are parasitic, that means they can cause disease. Example, Plasmodium that causes malaria. Next, they live in water, damp soil, and in other organisms. That is their habitat. Next is that they move by cilia, flagellum, or pseudopodia. Some of them don't even have any part that they use for movement, as we have in Plasmodium. They don't have any organ that they use for movement, but some of them they move by cilia, flagellum, or pseudopodia. As we are going to discuss some of these organisms that move using this part that we have just named. So let's start with the first example here, amoeba. But mind you, those characteristics you're supposed to know them, because whenever they are going to ask you, they will start by asking you to name the kingdom of this organism that you have identified, and you will be asked to give two or more reasons why you stated that it belongs to this kingdom. So in that case, you have to give the characteristics. So this amoeba, it is having an irregular shape. That means it doesn't have a definite shape. This amoeba, it is a unicellular microscopic organism. It is found in water and moist environments. In case you will be asked to give it habitat, it is found in water and moist environment. So based on classification, amoeba is being classified under the kingdom protista, under the phylum sarcodyna or protozoan. This protozoan, it is most often used in older classification. In modern classification, it belongs to the phylum sarcodyna, but in older classification, they put it under the phylum protozoa, and it belongs to the class Rhizopoda. Example of this species we have is Amoeba proteus. Now let's talk about the characteristics of phylum sarcodyna. So number one, they move by using pseudopodia. Pseudo means false. The podia is talking about leg. So in this case, they have false leg. How they move? When they want to move, they can form the pseudopodium in the direction in which they want to move. So if they want to move this way, they will first cause a protrusion or elongation of the plasma membrane. When the plasma membrane is being elongated or protruded, the cytoplasmic strand will move into it, As the cytoplasmic strand move into it, the body at the back will retract and move in that direction. So that is how they move by using their pseudopodium. And the pseudopodia is a plural form of the pseudopodium. Number two, they are unicellular and microscopic. What does that mean? It means that we cannot see them with our naked eyes except with the use of microscope. The micro means small, scope is talking about view. So we can view those small objects by using an instrument we call microscope. Number three is that they reproduce mainly by binary fission. When they want to undergo binary fission, first they will divide the nucleus into two. That one nucleus they are having, they will divide it into two because it is the nucleus that contains the genetic material. The nucleus will move to opposite pole. This one will move at this part, while the other one will move at the other part, and they will constrict at the middle. When they constrict at the middle, they will cut from that point, and at the end of the day, we will have two daughter cells, each one having its own nucleus, and each one undergoing all the life processes as it should be. Next, under the characteristics for phylum Sarcodina is that they feed by engulfing food particles through phagocytosis. When we talk about phagocytosis, we are talking about the taking in of solid particle by forming a vacuum. And we have pinocytosis, which is almost the same as phagocytosis, but in pinocytosis we are not taking in solid particle, we are taking in liquid particle. That is the difference between phagocytosis and pinocytosis. But here we are going to talk about phagocytosis. So when amoeba want to take in food, as we have on this slide here, it will first protrude its pseudopodia in order to surround the food. When it surround the food, this pseudopodia here will cut from this point here in order to form a vacuum. Enzymes will be secreted in order to digest the food. When the food is being digested, the food is being absorbed. When the food is being absorbed into the cytoplasm, this food will be assimilated into the cells of the amoeba. The body cells of the amoeba will make use of it. And what will happen? The waste or the undigested food particle in that membrane, it will move closer to the plasma membrane of the amoeba, and it will open up in order to expel the undigested food particle outside of the amoeba. Next we have here on the slide is osmoregulation in amoeba, how the amoeba regulates its water. So we are going to start from number one here. Water enters due to osmosis. As we define osmosis, it is the movement of water molecules from the region of high water potential to the region of lower water potential across a semi-permeable membrane until the molecules are evenly distributed. So water enters due to osmosis. When more water enters into the amoeba, it may start to affect the normal functioning of the amoeba. So what will happen? The excess water will enter into the contractile vacuum of the amoeba so that they will not affect the normal functioning of the amoeba. This contractile vacuum will swell up. When the contractile vacuum is full, it will move closer to the plasma membrane of the amoeba, as we have here in 4, that the contractile vacuum moves to edge of the cell. So when this contractile vacuum move closer to the plasma membrane of the amoeba, the contractile vacuum will burst and expel water, as we have here in 6. After the contractile vacuum have expelled the water outside, the whole process or the cycle can repeat again. Next on our slide is another example of organism under the kingdom protista, that is paramecium. This paramecium is a unicellular organism. It is microscopic as I have explained it before, but this one it is having a slippers shape. Unlike amoeba that is irregular, that is not having a definite shape, in here it is now having a definite shape, it is having a slippers shape. Now let's talk about the classification of paramecium. It is found under the kingdom protista, just like amoeba, but under the phylum Xylophora, or you can call it Ciliata. It is found under the class Ciliaceae, under the genus Paramecium. An example of species here include we have Paramecium cordatum, we have Paramecium aurelia. You have to know their pronunciation and their spelling as well. But their spelling is more important because you will not be having oral exam but a written exam. On this slide here, we have diagram of a paramecium, and you can pause this video and know how to label it before you move, or you can move on and come back later, or you can learn it anywhere on how to label paramecium. Let's talk about the key features of this paramecium. One, it has the cilia. This tiny projection you are seeing coming from the pellicle is what we call cilia. The cilia is an ear-like structure that is used for movement and feeding, because it is the cilia that beat the food in order to get into the gullet. Next we have is the pellicle, the flexible outer covering that is used for protection. For them, they don't have the plasma membrane. That plasma membrane is being named as pellicle. They have the oral groove, and this oral groove it is used for the intake of food particles. These food particles are being taken into the cytosol where they are being digested. And we have the contractile vacuum. The contractile vacuum, it helps to remove excess water, thereby helping in the process of osmoregulation. That means the water balance of the paramecium. And we have two nucleus in here. We have the macro nucleus and we have the micro nucleus. Macro means large, so the larger nucleus here is what we call the macro nucleus, also called the mega nucleus. And we have the micro nucleus. Micro means small. So the smaller one here, this one here, is what we call the micro nucleus. And the micronucleus, it is involved in reproduction, while the macronucleus, it is involved in metabolic activities. Number 7, number 8 here, production that the paramecium undergoes. One is the binary fission, that is the asexual mode of reproduction in paramecium, and we have conjugation, that is the sexual mode of reproduction in paramecium. But we are going to discuss them in detail later. So let's start with the binary fission, that is the asexual reproduction in paramecium, how it occurs. So when paramecium want to undergo binary fission, similar to what we have discussed in amoeba, but this one here, it is having two nuclei. It is having the macronucleus and it's having the micronucleus. So in here, the macronucleus will divide by mitosis, while the micronucleus will divide by meiosis. So after the macronucleus and micronucleus have divided, they will move to opposite pole. When they move to opposite pole, as we have it here, the oral groove will also be formed at another end. So at the end of the day, we have one macronucleus, one micronucleus, and one oral groove at both ends. And the paramecium will constrict at the middle and it will cut from that point. At the end of the day, we have two daughter cells of paramecium as we have it on this slide. We have one at the upper part and one at the bottom part. So that is how binary fission occurs in paramecium. Now let's talk about conjugation, how it occurs in paramecium. You have to listen and listen attentively because in here we have many things that has to come up. So let's start with A. Two sets of compatible types, that mean paramecium of the same species, will lie side by side and partially fuse. That means just a little part will be fused, not all of it will be fused. And we have in B that meiosis of micronuclei will produce four haploid micronuclei in each cell. So this micronucleus here will divide to give us four haploid cells, and the other one here will divide to give us four haploid cells. Whenever meiosis occurs, four haploid cells will be produced. So the micronucleus that is responsible for reproduction will undergo meiosis. And when that occurs, four haploid cells will be produced. But mind you, in here, the macronucleus, the larger nucleus I mean, will not be divided. It is only the micronucleus, the one that is responsible for reproduction, will divide because it is the one that has the genetic material and it is the one that is responsible for reproduction. In C here, three of these micronuclei in each cell will disintegrate. That means three of these haploid micronuclei in each cell will disappear. As we said, when that one undergoes meiosis, it was being divided into four. Among these four, three of them will disappear. Only one will remain, bringing us back to just like what we have in A. Now in D, what will happen? The remaining micronucleus in each cell will divide by mitosis. This time not meiosis but by mitosis. By mitosis, when a cell divide, it will give us two. When two of them divide, they will give us four. When the four of them divide, they will give us eight. So this one cell here, the remaining one, will divide by mitosis and it will give us two diploid cells. These two nuclei, one of them is called stationary nucleus. That means it will just be at one position. The other one is called migratory nucleus. That means it has to move. It has to migrate. So what will happen as we have in E here, that the cells swap one micronucleus. So the migrating here will move to this point, and the other migrating here will move to this point. So at the end of the day, you can see there is a mix of color here. We have green and red. But before now here, we are just having green, and here we are having red. But when these migrating nucleus, when they swap, what will happen? We have this mixed color. We have the green, we have the red. This is just for explanation. I'm mentioning this column. It is not like the stationary nucleus here is green or the migrating nucleus here is red. After the cells have swapped their nucleus, they move apart. When they move apart, each one of them will undergo these processes that we are going to explain. This one will undergo the processes that we are going to explain, and this one will undergo the same processes that we are going to explain. But we are going to focus on one, and we are going to focus on this one that is part of the cycle. So after they have separated, the two micronuclei will fuse to form one. That means the migrating nucleus and the stationary nucleus, they will fuse to form one nucleus. When they fuse to form one nucleus, the one nucleus will undergo 3 rounds of mitosis to produce 8 micronuclei. Just imagine one undergo mitosis to give us 2, these 2 undergo mitosis to give us 4, these 4 undergo mitosis to give us 8. That is how we have the 8 micronuclei. So after we have got the 8 micronuclei, as we have it here in I, that 4 micronuclei will become macronuclei, and the original micronucleus will now disintegrate. So after that, we will have 4 micronuclei and 4 macronuclei. So in here now we have our macronuclei and we have our micronuclei. So what will happen later? This cell here will undergo 2 rounds of binary fission to give us 4 daughter cells. As we have it here in J, you can see the 4 daughter cells here. So how we have the 4 daughter cells from the 2 rounds of binary fission? The 1 will divide to give us 2, the 2 divide to give us 4. So that is how the 2 rounds of binary fission will give us the 4 daughter cells. And don't forget, I said here in E that the cell will swap their nucleus. And don't forget, in F, I said the mating cells will separate after they have swapped their nucleus. And I said we'll just follow with one and explain what will happen. And I said the same thing will happen to the other one. And mind you, when we have explained this one, we said it will give us 4 daughter cells. The other one as well, which we didn't explain, will give us 4 daughter cells as well. So at the end of the day, these 4 and the other 4 will give us 8 daughter cells. So in conjugation, at the end of the process, it will give us 8 daughter cells. Now let's talk about osmoregulation in paramecium. How osmoregulation occurs in paramecium. We are going to start from the bottom here. We have the empty vacuum. As more water enters into the paramecium by osmosis, this water may tend to affect the normal functioning of the paramecium. So in that case, what will happen? The radial canal will take up the excess water from the surrounding cytoplasm, and this water will move from the radial canal into the vacuum. This thing you are seeing here, these are the radial canal here, and the round structure you are seeing, in which the radial canals are surrounding, is what we call the vacuum. So this vacuum, after it has taken a lot of water, it will become full. When the vacuum becomes full, it will move closer to the pellicle. When the vacuum moves closer to the pellicle, it blends itself with the pellicle and a pore will open and the vacuum will contract. When the vacuum contracts, it will expel the water out from the cell into the environment. So that is how paramecium regulates its water balance. So that is how paramecium undergoes osmoregulation. If you don't understand this part fully, as I was going fast, you can pause, copy the notes on the screen and read it. It can help you a lot as well. Next we have here on the slide is euglena. Now let's talk about the classification of euglena as we have done with the previous examples that we have discussed. Euglena is placed under the kingdom protista, just like any other examples that we have discussed before, but it is placed under the phylum Euglenophyta. In older scheme of classification, it is placed under the phylum Mastigophora, and it is placed under the class Euglenoidae. Mind you, you're supposed to know this spelling. You're supposed to know the spelling of euglenoidae. So this is the diagram of an euglena. So you have to pause and learn how to label this diagram correctly. We have the eye spot that is used for sensitivity. We have the flagellum that is used for movement. We have the gullet for passage of food. We have the pellicle for protection. We have chloroplast or stellate chloroplast for photosynthesis. We have the nucleus that contains the genetic material, and we have the cytoplasm that provides the site of chemical reaction, and it is in the cytoplasm that the organelles are being suspended or are being held in place. We also have the contractile vacuum that is there for osmoregulation, and we have the paramyelin granule that is there to store starch. These are the main parts that you're supposed to know together with their functions. But please, mind you, you're supposed to know how to label it correctly. I know you can identify it, but please know how to label it and know their functions. This euglena is having an animal-like characteristics and plant-like characteristics. We are going to discuss both. One, it has an eye spot for vision or sensitivity. It has flagellum for movement. It has contractile vacuum for osmoregulation, and its body is covered by pellicle. You know, plant is being covered or is being surrounded by cell wall, but in here the body is being covered by pellicle. So as we have informed that the body is covered by pellicle instead of cell wall, and it has gullet, the passage for food. The last one, it can consume food by phagocytosis. That means it can ingest solid food by forming a vacuum. Next on our slide is the plant-like characteristics of an euglena. What are some of these characteristics that makes an euglena to be like a plant or to be placed under the kingdom planting? One One, it contains chloroplast for photosynthesis. It is only plants that are having chloroplast which they use for photosynthesis. Chloroplast is one key defining feature of organisms under the kingdom Plantae. So if a euglena is having chloroplast, that means it is having a plant-like characteristics. Two, it has chlorophyll molecule which makes it green like the leaf of a plant. But the one and these two, they are just the same thing. You can just take one. The second one is that it stores excess glucose in the form of starch. Plants store their glucose in the form of starch, while animals store their glucose in the form of glycogen, not in the form of starch. And the last one is that it has paranylon granules which stores starch.